Dear attendees, welcome to the current session of Bioinfo course. I'm Sema Yoshiri. Dr. Christopher Mayack will soon deliver a speech under the title of Environmental Exposures that Can Accurately Predict Honeybee Infections. And at the end of the speech, we will have a Q&A part.
Okay, thanks very much for the introduction. Today I will share with you some recent results that we've obtained that focus on improving honeybee health. And this is important because as you can see here from this image, the honeybee is an integral part now of our food production system for the mass production of crops due to their pollination services. So their decline in health on a global scale is of concern because they are playing a critical role in getting food from the farm to fork or from farm to table due to these pollination services to increase not only the quality but also the yield from the crops that are grown by the farmers. So as I mentioned before, the bees are critical for not only food production but also the economy in general. So the bees pollinate about 1/3 of the food that we eat every day. So a lot of the fruits, nuts, vegetables that we enjoy are either partially pollinated by honeybees or sometimes are nearly completely dependent upon honeybees for pollination. So one example of this is almonds are 99.9% dependent upon honeybee pollination. And that's in California, where they produce a lot of the almonds for supply around the world. And then, of course, you're probably familiar with honeybee products. So these include honey pollen, propolis, bee venom, royal jelly, and beeswax. We make use out of these as well. So if their health or numbers are to decline, this would affect the production of these as well. And roughly it's worth about 150 billion euro worldwide, or about 10% of the market value of food that the bees pollination services provide. And interestingly enough, Turkey is home to the largest number of beekeepers per capita in the world. So for Turkey in particular, keeping bees healthy are critical for many of the beekeepers that are here in Turkey as well. So as you can see, this is an issue in Turkey, but also around the world. So these orange countries here highlighted are countries that have suffered from the most current decline in bee health. So you can see it's nearly a global phenomenon. And over the last years, this has been studied in detail in terms of losses, winter losses and summer losses. And what you see from this figure here is that the losses are at an unsustainable rate. So what's acceptable to a beekeeper is about 15% losses of their beehives. So they expect some sort of loss each year. And you can see on average, we have about 30 to 40% losses since about 2006, when this most recent bee health decline was identified. So there's now a concern that we may not have enough bees to meet the pollination services that are in demand for increasing crop production. And the decline of bee health has been identified due to multiple factors that are environmental in nature. So we have new parasitic exposure, exotic diseases, poor beekeeping management practices, weather patterns such as climate change, poor nutrition, queen failure, so poor quality queens, genetic weakness due to a small genetic pool, or not enough genetic variation for the bee to be able to adapt to these different stressors, and then, of course, pesticide exposure as well. So what this highlights is that the bee decline is multifactorial, so that means it's a complex problem, and as such, a systems biology approach, which is what I'll show you in this talk here, is suitable for determining, how these factors may interact to cause a synergistic decline in bee health. So what we have here is the overall decline of the number of hives being managed. So this is data from the United States, but there's a similar trend around the world as well. And so the most recent decline in bee health is nothing new. So bee health or bee numbers have been declining for quite some time. And that's especially true when the varroa mite, which is a parasitic mite that's very damaging to bees, was introduced to the beehives in the US around the 1980s or so. And in this most recent bee decline health, there's this what's called colony collapse disorder, which is a relatively new phenomenon where bees go out of the hive and they don't return. So they're disappearing from the hive. And from this most recent decline in bee health, what we see are the states here being affected, and there's no clear, really striking epidemiological patterns to this. So again, it was hard to identify what the exact causes are for this decline. But it has been narrowed down to these major factors shown here. So pathogens or disease, pesticides, poor nutrition, and climate change seem to be the major players. And I'm going to focus on disease and pesticides, so these stressors in particular, on the research that I'll present today. So even within diseases and pesticides, there's many different kinds of diseases that can infect the bee and have been attributed to their health decline. So there's different fungal pathogens. You can see here viral pathogens. There's probably many more. There's 13 different kinds of viruses that we can actually identify and measure. And then we have bacterial infections such as American and European foul brood, and then the varroa mites, which is actually an ectoparasite living on the outside of the bee that feeds on the bee hemolymph and fat body that can vector viral infections as well. So they're quite damaging to bee health. And then within pesticides, we have different classes based on their target, right? So we have fungicides, herbicides, miticides, larvicides that we use, and then insecticides, of course, that are used in a lot of agricultural settings. They're also used for ornamental plants as well in suburban, urban areas. So they're quite pervasive and they're consistently put into the environment, and some of them take a while to break down. So we have many different insecticides, and the most recent ones are these neonicotinoids that have been linked to the recent bee health declines. And so as you can see, even within these two stressor categories, there's many different potential pesticide exposures and diseases that they can suffer from. And so this really led to a new initiative started back in 2015 when I was at Swarthmore College with these individuals here. So Brad Brodrip, Helen White, and Anthony Mascherone. And the project that we wanted to start stems from quantitative genetics, where we know that the phenotype is equal to the genotype plus environment. So what this means is both genetic and environmental factors can influence a phenotype. In this case, the phenotype of interest is bee health. And what's been used many times to characterize the effects or certain predictors of the genotype is transcriptomics. So looking at gene expression profiles and seeing how those are associated with, say, a diseased phenotype or an unhealthy phenotype. And what's been missing over the years is more of the environmental component in studying how that combines with transcriptomics or genetic factors and describing or characterizing predictors of this phenotype. So exposomics attempts to capture or account for this environmental component. And the idea is measuring all the exposures, both endogenous and exogenous, that the bee is exposed to. So those are external and internal changes in exposures or chemicals. So one example would be metabolites. So this includes metabolomics as well as the chemical, immediate chemical environment that the bee might be exposed to, which may include pesticides. And so what we have here is below is an illustration of the exposomics paradigm. And you can see here, as humans, as we develop through time and throughout our lifetime, we have a number of different exposures These exposures can include medicines we're taking, our diets, exercise, the environment that we live in, stress, and that is going to have an effect on the phenotype or the individual in the phenotype output that we see down here that we're interested in measuring, which is the health of the individual. So with the exposomics, it's capturing also changes internally. And so here is showing metabolic pathways. And so with these metabolic pathways, that might be a cause of the external exposures. Changes in that can also be used as a predictor of what might be leading to changes in health or the phenotype. So that's the idea of the exposomics paradigm being used to determine environmental predictors of health. And the idea we had was, with the systems biology approach, applying this exposomics to bee health, and also using the systems biology approach that's been used in the medical field, applying that instead of to humans, to bees instead. So what we have here, is these are the typical inputs you would have from a biomedical study using systems biology. So you have a bunch of data that's going in, perhaps from model organisms, from clinical data, and other measures to characterize the phenotype. And then there's measures, usually omics, that are used, so proteomics or transcriptomics, to determine how the biology is being affected, and how these genes and proteins, how's their expression being associated with the phenotype. And in order to accomplish that, we use bioinformatic statistical tools and to integrate and analyze the interactions and the associations. And so the goal from the systems biology approach is to hopefully develop novel biomarkers for rapid diagnostic of the disease, maybe establish actually a pattern of biomarkers that might be more accurate in diagnosing a disease, understand the pathogenic molecular pathways or models that might be responsible for causing the symptoms of the disease. And this also includes changes in metabolic pathways or biochemical pathways. And then from this, novel therapeutic drug targets might be able to be developed if we understand the underlying mechanisms responsible for producing the disease phenotype. So our idea here is, again, applying this to bees. So in this case, our inputs are going to be healthier and unhealthy beehives, or certain diseases that the beehives have. And adding to this approach, we've included exposomics as well, where we're measuring the external environment exposures to the foraging honeybees and the in-hive bees, and integrating it with these other analyses. So the overall workflow here of our approach in detail is shown here. So the idea is that the bee, if it's unhealthy, is likely to have a number of exposures or pesticides, perhaps has some diseases as well. And we're going to see if there's any association between the two. And the goal is to see if we can come up with chemical biomarkers for predicting disease or pesticide exposure. So this involves, honeybee hive sampling. From that sampling, we use gas chromatography, QTOF analysis. So this is high resolution gas chromatography analysis. And with this mass spec data, we can identify the chemical exposures in totality that the bees are exposed to. This also involves, instead of using transcriptomics, in this case, the omic data is integrated with the disease, so the presence and abundance of the different diseases I mentioned before. And we use qPCR and PCR methods to screen for these common bee diseases. And then finally, we did an R analysis and generated, in this case, a correlation matrix to draw these significant associations across the diseases, as well as finding these associations with the chemical exposures. So from this, we were able to identify some chemical biomarkers that are highly associated with and significantly associated with the presence of these diseases at the colony level. So the sampling involved hives across the urban, suburban, and rural gradient, and that's shown here. So they were sampled in Philadelphia for the urban area, and then outside of Philadelphia for suburban and rural sampling sites. So there's 10 different apiaries that were used. 30 hives were sampled in total for this first study, and then bees were pooled together for the disease screening, and then these pooled bees were also used, macerated and used for the LC-GC-QTOF analysis to generate the exposome. So that's the overall outline of the study. And a follow-up study, which I'll show you, is we sampled these similar sites, these 30 hives over a 3-month time period as well, that included our longitudinal sampling design. So there's 2 studies. I'll show you the results from both. And in both of these studies, we screened for these diseases that I showed you before, and these are the most common diseases known to affect bee health. And the output from the QTOF analysis is shown here. So this is 30 hives that are overlaid, and here we have the different peaks representing different chemicals. So the entire hive profile is here. And in total, we were able to identify and annotate about 1,723 exposures in the first study and 4,631 exposures in the second. And this was generated from GC and then LC-QTOF, respectively. And we were able to identify in the first study the 20 most common pesticide exposures, and then we analyzed these in detail. So these 20 pesticide exposures were found relatively high incidence across all 30 hives analyzed. So what you see from this profile here is that there's a lot of similarities, but there's also some key differences. And with that variation, we might be able to find associations with a change in profile with the incidence of a disease being in that hive. So if we look at this at a broad level, and we use unsupervised hierarchical clustering, you can see just if we were, as a beekeeper, to say classify our hive as unhealthy, or maybe we don't know the status, we're not sure. If we record that the hive is collapsed, and we can compare that with a healthy hive, you can see just generally with this heat map here showing relatively higher abundance of the chemicals and then relatively lower abundance of chemicals in blue. These are ranked as the top chemicals with the largest change across these classifiers is shown at the top, and then it goes down from there. So what we see in general here is quite different profiles, which is encouraging sign that we might be able to identify chemical biomarkers or chemical biomarker library that will enable us to eventually classify whether a hive is unhealthy, healthy, or perhaps even collapsed. So we have a lot of changes in relative abundance overall in the hive exposomes. So if we look at it in a bit more detail, this is in association with a particular disease, a fungal pathogen, nosema, that we looked at in detail. And you can see as we go from right to left, in general, the chemical entities, the chemicals as part of the exposome, have very different profiles. And as we go from right to left, you can see more dysregulation in these profiles in comparison to the healthy hives or the uninfected hives. So this yellow color indicates a neutral change, a relatively little change in the hive chemical. So again, there appears to be some sort of association with the presence of disease and a change in the relative abundance of chemicals. So what was generated from this data is what's known as a health index that combines the disease abundance across the ones that I showed you before and the pesticides themselves. So if we look at the relationship between the two of them, we can see that the pesticides are generally driving this health index. And the idea was, with this health index, we could use it as a measure to indicate whether the hive is healthy or unhealthy. So it appears that pesticides are mainly driving whether or not, in general, if a hive is healthy or not. So that's in contrast to the disease. You can see it's a less strong of a relationship here with a lower R value with this correlation analysis as it relates to the health index. So the chemical biomarkers that we're able to identify are shown here in this table that are associated with a particular disease. So we're able all of the diseases together, for example, and create a disease index. And with that, we have particular chemical biomarkers that are highly associated with this. And this is different from the other specific diseases that we see here. So this is promising, where we may have biomarkers that are useful for just indicating whether a hive is infected or not with any sort of disease. And then we have some that are more specific, like shown up here with Lake Sinai virus. This chemical biomarker could be used for predicting whether or not the hive is infected with this in particular. So these are the top hits identified with that analysis. And we decided to take it a step further and look at the relationship as it's plotted in these different metabolic pathways. So this is derived from the KEGG database. And what we see here is that in general, the chemicals are found in relatively higher abundance, which is indicated by the green color. And you can see that linoleic acid, for example, is highly associated with this disease index. And this is an interesting finding because linoleic acid here, which seems to be upregulated, tends to have antimicrobial activity and effects. And we can see here that this tetradecanoic acid is associated with Nosema cerana infection, and this corresponds with other literature findings showing that lipid or fatty acid metabolism is dysregulated in these infected individuals. So this is where we see certain metabolic pathways being affected, and perhaps some of these exposures are affecting, say, the same pathway, and this is where we might see a synergistic decline in bee health. So we were able to analyze the second study in a bit different way, which involved a little bit more sophisticated bioinformatic tools. And so this involved pooling the hives together to have a bigger sample size. So the 30 hives sampled over 3 time points were pooled together for a bigger data set. And then with both the exposome and the disease screening data, these were combined and were pre-processed and normalized. And we classified the hives as just being infected or not infected to simplify the analysis due to our low sample size. And then with each disease, associations were identified using PLS analysis, partial least squares analysis. And from this, we were able to actually identify a set of biomarkers. So a library of biomarkers or features. And with this, we did machine learning analysis to validate these biomarkers. So we had a training set and a test set, and we used these different algorithms here to run a threefold cross-validation, and we did that twice. And this was able to validate the precision and accuracy of the chemical biomarker libraries for each one of these diseases here. And what you see here is pretty promising results, okay, based on the AUC values. So this incorporates the sensitivity, the accuracy and sensitivity of the biochemical library for predicting whether or not a hive is infected with, in this case, acute bee paralysis virus or Kashmir bee virus or Israeli acute paralysis virus. So this is part of the same complex. It's relatively similar. And you can see this is relatively consistent across the different diseases. So these are viral infections here, different viral infections. And you can see we have at least 90% sensitivity and accuracy. So we did other measures too recently. And so we have precision, and recall is also around, on average, 90% or so. These are some of the better results from the different algorithms that we used. Here are two more viruses. Again, very high levels of sensitivity and accuracy. And this is also true for bacterial infections such as European foul brood, Nosema serrana, which is a major fungal pathogen globally of bees, and also of varroa mites in particular. So we were quite excited about these results. And a summary of the results are shown here, where again, across the different validation methods, you can see the average here is around 90% or so. And that holds true for across the diseases as well. And what you see here on the right is the number of biomarkers. So this is interesting. So to have this kind of accuracy and sensitivity and precision, only 36 biomarkers, chemicals were needed for these viral infections. But if we go to sac brood virus, for example, 398 is the number of biomarkers that are needed for this kind of accuracy to be able to predict if the hive is infected with this or not. So we do see here that there are certain chemical biomarkers that were identified in those libraries that are common to all of them. So these might be biomarkers that could be used again just to see if the hive is infected with any of these viruses. So this is looking across the different viruses that affect bee health. And you can see some are unique, completely unique to the virus, and some are shared by, you know, two or more viruses as well. So there's quite a bit of variation that we see. And the same is true for the non-viral infections. So surprisingly, these are quite different parasites in terms of their life cycle and lifestyles, but nonetheless, there's certain chemical biomarkers that are common for all three of these or shared by two of these. And then there's some that are unique as well. So again, that gives promise that we might have both general and specific biomarkers potentially for these diseases. So these really need to be now validated in the laboratory, either in vitro or in vivo, to establish a cause and effect relationship. And then once we've established that, then we can have much more confidence in them being truly reliable biomarkers for predicting bee diseases. So the idea behind this was in part not to sacrifice any of the bees in order to sample or order to determine whether or not the hive is infected with a disease. So what we've done before in the past is developed a passive sampling method, which involves using a silicone wristband that can be placed into the hive for 24 hours. And then it can passively absorb these chemicals that are in the hive environment, and then later extracted in the laboratory and analyzed with GCMS like we've done before. And we were able to find some of the top hits from our first study within these silicone bands using this technique. So these 3 chemical biomarkers were able to be captured passively. So this is relatively easy for the beekeeper. They don't really need to open up the hive. They don't need to sacrifice any bees. And then the idea behind this is to standardize this process and have the band sent to, say, some centralized bee health center that screens for these chemicals in a high throughput manner and can aid in giving the beekeeper a rapid diagnosis of what's infecting their hive. So the main problem now for beekeepers is they have very poor diagnostic tools for detecting the bee diseases, and they're too costly and take too much time. By the time they figure out what their hive is infected with, they don't have time to alter their management practices to try to improve the bee health. So that's some of the goals we have with these chemical biomarkers. So some future research directions include validating the top biomarkers experimentally, empirically, in vitro and vivo, and then optimize this passive sampling method to make it even quicker and cheaper and easier for beekeepers to capture these chemical biomarkers that we've identified. And then lastly, an idea is to integrate these chemical biomarkers with other data that's continuously being connected with beehive monitoring systems that use IoT technology to continuously record temperature, humidity, pressure, and sound, which has already been established. And if we can draw associations with that, with our chemical biomarkers, we might be able to use this data to be able to indirectly predict if a beehive is sick, and if it is, what is it sick with? What particular disease does it have? And with this technology, it can inform the beekeeper of the hive health problems, in theory in real time, because it's connected to a smartphone application. And the beekeeper could learn, what disease is infecting their hive in real time. So with that, I'd just like to thank some of my students that were involved in this study from Swarthmore College that I work with, as well as Rob Brodrup and Helen White and Anthony Mastrone. And then also the Sabanci University students shown here, undergraduates and graduate student Mohammed here for conducting the analysis. And last but not least, Agilent and Tubitech for their financial support for the studies that you saw here. So with that, I thank you for your time, and I'll be happy to take any questions.
Thank you for your presentation, Dr. Mayack.
Mm-hmm.
Then we can proceed with the Q&A part. First of all, I would like to start with the questions prepared by our BioInformatics team.
Mm-hmm.
And the first question is, as we know, in different geography, we have different honey products depending on tree or pollen types. What type of change we should expect for the honey products in the changed ecosystem because of climate emergency?
Mm-hmm. So just to rephrase, so the question is in relation to yeah, what kind of changes we might see in honeybee products due to climate change. Is that right?
Yeah.
Yeah. So, well, it's the honey and pollen and and propolis are dependent upon flowers or tree resin. And the issue with climate change is what's known as what they call a phenology mismatch. And so that with the climate change, there may be flowers that, say, bloom earlier or later because the climate has changed. And the flowers may be able to adapt to the change, say, better than the bees. And therefore, it's been shifted in terms of when they'll bloom. And the bees may not be active then, may not have come out of hibernation, for example, yet from the wintertime. And so that's going to cause some problems with foraging efficiency and, going to make it more difficult for the bees to find the flowers. And that's where they get the nectar and the pollen, and the nectar eventually becomes the honey, and the pollen is just consumed directly. And then same is true with the trees and for tree resin that will eventually become propolis. So those are the bee products that will be affected most, say in comparison to bee venom, or I guess you could even argue beeswax might be affected, because if the bees can't get sufficient, honey or food, or in general poor nutrition, it's been shown to make them more susceptible to pathogen infections because of a suppressed immune system. And when their energy is taken away from these infections, that they have more difficult time in producing beeswax because it's very energetically expensive. So it's all connected to food supply for the bee, and having the energy to fight infections and produce the beeswax. So that can be affected as well in royal jelly production. So really, all of it, to a certain extent, will be affected.
Thank you for your answer. And the next question is, how important are social networks within and outside the honeybee colony for infections?
Mm-hmm. Very important. They are highly social, so they have many interactions, and not only do they come in contact with one another, but they also share food too. So many of the diseases are thought to be shared through sharing the food, and bees have been shown to have evolved to have a particular social network to protect the more valuable members, which are considered to be the younger bees, because eventually they'll get more work out of those bees. So if an old bee dies, it's not as costly as having a young bee die. And that's not as costly as having the queen die, which is the most valuable member of the colony. So they're arranged such that the interactions are limited. So the older bees tend to be on the outside and interact with one another. And that's also true for the inner bees as well. So their social structure and interaction can both be beneficial to prevent getting infections, their social behavior itself, and then also it can lead to higher transmission for sure within a colony. And generally that's what we see is once a disease gets in a colony, it can spread quite quickly because there's a lot of individuals. They're genetically similar, so they have the same sorts of susceptibilities, and they're living in a contained area inside the hive, and it's very densely populated. So, and we know from COVID, right, with social distancing and how key that is, the bees aren't doing that. They're interacting all the time. Now, you asked about outside the hive. The main sort of transmission route thought to be taking place outside the hive is from shared flower use and from sharing the same nectar source, for example, or even coming in contact with these pathogens from defecation on flowers as well. So that's really what's thought to be the major horizontal transmission routes outside the hive. But you also have some studies showing these deformed wing virus, for example, being found in ants and yellow jackets. And these yellow jackets enter the hive and try to take the nectar and around the fall time, around now. And they're entering the hive. So they could be a potential reservoir or a source of transmission as well. So if you think about it at a community level, there's other species in the environment that may be potentially interacting with bees. So ants will go into beehives too, to again try to take some of the nectar or honey that's there. And they could potentially transmit the deformed wing virus too. So we don't know for sure, but it's definitely plausible. So those are some examples of potential transmission routes due to the social interactions or interactions with other species outside the hive.
Thank you for your answer. I'm reading the next one. The pesticides used cause susceptibility to infections in bees. Can foresight studies be conducted to unify agricultural products and bee health initiatives at the same point?
Unify agricultural products. Yeah, so it is true that pesticides do cause increased susceptibility to infections in bees, and some studies have shown that pretty clearly. And also, pesticides and parasites together can increase the mortality synergistically. So I think this touches on the idea of having a bee health center, I think, where beekeeping practices could be more standardized and beekeepers could be better informed of the diseases or pesticide exposures that they have in their hives. So there are some governments and government agencies that screen for this on a regular basis, but I don't think, unfortunately, there's not a lot of or enough communication, I would say, between the research groups and government organizations and their findings, and communicating that to the beekeepers and what they should be doing to as good practices to treat the diseases or avoid exposure to the pesticides. So there's could be better crosstalk between the agencies, and that will start to then unify agricultural products and I think bee health initiatives. So I'm not sure what's meant by foresight studies. I guess maybe studies that could predict and how that relates to unifying the agricultural products. But that's kind of how I see it or how I view it. So I hope I answered the question okay.
I think they mean the studies ahead.
The studies ahead. Yeah. Well, there is this what's called One Health Initiative or One Health Perspective to bee health, which is trying to now look at bee health more holistically. And what's emerged from that is biomarkers being a central tool in assessing bee health, because they can be used rapidly and then they can also potentially be used across bee species too. So honeybees are in decline, but also wild bees and bees in general are in decline, rapid decline too, right now. And so to improve health of bees in general, biomarkers seems to be a good start, or that's what's been reported recently in some review papers. So that's kind of a unified goal right now. And then also developing vaccines for some of the bee viruses seems to be a concentrated effort right now to help with increasing, agricultural productivity.
Thank you for your answer. And the next question is, can airborne organic pollutants be detected using honeybees as bioindicator organisms? Can such study be done?
Yeah, so this ties into this work too, which I didn't cover that much, didn't really talk about at all. But old studies have shown or demonstrated that honeybees, because they forage out about a 10-kilometer radius from their hive, can be used what is known as environmental sentinels. So they go out and in this large area, and they're getting exposures to pollutants and pesticides, and so they can be used as monitors for the environment. And so with this sort of analysis, you could look for particular chemicals with this exposome paradigm and learn about how healthy the environment is, the ecosystem is that surrounds the hive. So bees can be used as bioindicators of the environment, the surrounding environment. That's been done a little bit, but there's I think new excitement to do that some more in the near future.
Thank you for your answer. And the next question is, how successful are currently used simulation models such as Beehive and EPISRAM in accurately predicting honeybee infections?
So models, so if you're referring to mathematical models, simulations have been created, and if you look again at the social network aspect, they've been used and have been useful tools in understanding routes of transmission and how diseases spread through a social network. So they've been used in that sense. And some models have been used to understand how a colony can eventually collapse too, from certain mortality rates, from certain stressors. So modeling, mathematical modeling is used quite a fair amount in honeybee studies, and it mostly stems from trying to understand the social complexity and what that means for the survival of the individual, and also the survival as a colony as a whole. So looking at those dynamics, with modeling has been a useful tool. So it's definitely applicable.
Thank you for your answer. Now an attendee just raised a question. For what reasons does the queen bee weaken? Are environmental factors effective? If so, what are these factors?
Yeah, so queen bee can be infected, for example. So studies have shown that they can be infected with deformed wing virus. They can be infected with nosema. They've demonstrated that. Queen bees will weaken naturally with age. They live to be about 2 to 3 years old, and so beekeepers do replace them on a regular basis. Sometimes a queen could be weakened if they don't get good nutrition. So what determines a queen bee being a queen bee instead of a worker is actually being fed a special diet of royal jelly. And so it's much more nutritious and higher energy than the other food that the other bees get when they're developing. So if they don't get a good diet of that royal jelly, so if the hive is weakened somehow and the bees are unable to provide that high quality royal jelly, then the bee won't develop into as good quality of a queen. You can think of queen bee being weakened in terms of reproduction if they don't have good mating with multiple drones. That's been shown to increase genetic variation and genetic vigor, is having multiple mating with the queens. And so that can be an issue too, where they're not mated well, they can run out of sperm, for example, and not be able to fertilize the eggs, which are needed to produce the worker bees, which are all female. Environmental factors, yeah, they can play a role because the queens are mated outside of the hive. They mate in midair in these drone congregations. And so if you have environmental factors that affect the drones getting there, drones can be infected too with diseases. These pesticides have been shown to affect semen quality in the drones. And so again, environmental factors too, the abiotic factors can affect the ability for these drone congregations to occur. The conditions have to be just right for this to occur, and it's not well known what the exact factors that need to be in place for a mating session to occur, a mating flight to occur. But environmental stressors during this process could weaken the honeybee queen and make it less likely that she's able to return back to the hive after she's mated.
Thank you for the answer. It was quite detailed. And the next question is, the bees that are infected with varroa are more likely to catch nosema. How is this possible?
So I know that the bees, infected or that have varroa definitely have a higher incidence of deformed wing virus. I'm not sure about nosema, but it could be the case. And how this could occur is a suppressed immune system from the initial infection. So varroa, for example, there's a few studies that show it can suppress the immune system. And also, recently, the varroa mites have been shown to feed directly on the fat body, so a lot of the lipids or fat stores of the bee. And lipids are important for energy supply for the immune system, in particular, lipid metabolism. And so, without any lipids, no good immune system either. And therefore, nosema can be able to infect the bee more easily. Yeah.
Thank you for your answer. And another attendee just raised the question, which environmental factors are effective in attacking the weak hive by the honeybees living in a strong hive?
Okay, yeah. I think I know. Yeah. So you have this sometimes. You have what's known as robbing. So stronger hives will overrun a weaker hive, especially in the fall time when they're trying to collect as much as possible. They hoard nectar to build up their honey stores so they have enough honey to get through the winter. So I'm not sure. I mean, the environmental factors would be definitely seasonal factors. So there's a lot of less forage available towards the fall time, and they go into more. They get more and more desperate to find food, So they get more exploratory, and then they start to then become attracted to going into beehives, and they'll fight each other, and the hive is weak enough, yeah, it can be completely overrun by a stronger hive, and the stronger hive's trying to get in there, not to take over the hive, but to get at the honey stores, and they'll actually uncap the honey and take all the honey. And yeah, this is known as robbing behavior, which is another way in which diseases can get exchanged. So you have a weak hive that's from infected, other bees going in there and then returning back to their own hive can spread those diseases.
Thank you for your answer. And I'm proceeding with the next one. Are there approaches to prevent the Nosema parasite from passing from migratory birds to honeybees?
Mm-hmm. Yeah, so there is it's a group from Spain that studies Nosema a lot, and have shown these, I can't remember the bird, I think maybe honey creeper or something, not a honey creeper. But yeah, there's this particular birds species that will eat bees, and the group in Spain have shown that this could be a potential way to spread Nosema long distances. So the bird would defecate out after eating an infected bee, and this defecation, they show, can land up on the top of honeybee hives, and then again bees can incidentally come into contact with this and spread the disease. So it sounds pretty amazing, but you have to keep in mind all it takes is one Nosema spore to get into one bee, and then from there can be millions of spores or hundreds of thousands of spores just from one bee, one individual. So once these parasites get in, they do a really good job of reproducing, replicating. So it's a system that's hard to study, and so they haven't really shown any direct evidence. But yes, this is true that in Spain they have a study showing that birds eating bees can defecate and potentially spread the Nosema parasite.
Thank you for your answer. We have another question from the YouTube chat. What are the effects of pesticides on the plants that honeybees feed on? Do these drugs make honeybees more susceptible to diseases?
Mm-hmm. Yeah, so the effects of the pesticides on the plant, if it's a desirable plant, the neonicotinoids are the newest class of pesticides that are used around the world, are coated in the seed actually of the plant. And as the plant grows, it exudes the pesticide. So it's what's known as being a systemic pesticide. It's throughout the plant because it gets the pesticide as it grows out of the seed. And consequently, a lot of the nectar and pollen can have low doses of that pesticide. So for the plant, it's good in theory because it reduces the pests, and so it should, kill the insects potentially feeding upon it. But it's bad for the plant if it needs to be pollinated by a pollinator, because it could potentially expose a pollinator to that pesticide. It's argued that these neonicotinoids are at such low doses that they won't really affect the beneficial insects, the pollinators, the honeybees. But what we do find is that these pesticides do make the bees more susceptible to diseases, and they definitely are at least a significant part of the bee health declines. And it's not too surprising because in these pesticides can be insecticides, but sometimes they're herbicides. So they're aiming to get rid of the weeds. But even the herbicides have been shown to have sublethal, so at very low doses, effects on bees' reproduction, physiology, behavior, and these effects have been shown to likely reduce, learning and memory, and so all these issues with survival and reproduction of the bee. So in the long term, they're having quite damaging effects. In the short term, the bee appears to be fine because it's not a lethal dose. So a lot of work has been done mostly on these sublethal effects, or the chronic, the more long-term effects on bee health.
Thank you for your answer. And the next question is, what is the relationship between the prevalence of Nosema disease and humidity in the air?
Mm-hmm. So Nosema and humidity, right?
Yeah.
I don't. So this is just anecdotal. So this is what my perspective is. There isn't. I don't think this has really been studied that I'm aware of, the connection between these two factors. But what I've noticed is if we have a cold, rainy spring, Nosema tends to be worse. I think humidity plays a pretty large factor actually in these diseases. In general, high humidity is bad for the host. It's better for disease transmission. Diseases are more likely to be able to survive outside of a host with higher humidity. So for viruses, bacteria, etc. And I think the same is true with Nosema. So yeah, generally when you have high humidity, I've noticed, or cold and rainy, where high humidity plus the bees are staying in the hive a lot, that the diseases seem to be worse.
Thank you for your answer. A question is raised from the chat. Are infections in honeybee only caused by biological agents? Is there different factors that might cause the infections?
Well, I guess, as with a strict definition of infection, I suppose it has to come from a biological agent, by definition, I think. But are there different factors that might ultimately cause infection? Absolutely. There's other stressors that we talked about, like poor nutrition or the pesticide exposure, or climate change that causes less flowers to be available, which then in turn, the bee doesn't have good nutrition, and then that affects the immune system, and then diseases can get in more. So I think bees can get sick still from other pollutants too. So we found some pollutants in the hives so it goes beyond pesticides. So there's other man-made pollutants that we found, and it's likely that they could make the bee sick in that sense. But I think, if you talk about an infection, it has to be caused by some sort of, pathogen or something like that.
Thank you for your answer. And another question is there a relationship between subgroups of honeybees? For example, Caucasians bees.
Got the Caucasian bees. Yeah, you got it. Yeah.
I'm sorry. Caucasian, I guess.
Yeah, you got it. Yeah. Yeah, the Caucasian bees, are in northeast of Turkey, and in a more mountainous area there up by Georgia. It's a special kind of subspecies. It's relatively rare. So to answer the question, yes, there's some studies that have shown this. My personal experience, I found there's a Russian subspecies of bees, and they tended to be fairly disease resistant. Or some beekeepers argue that the Carnolians are relatively disease have higher disease resistance in comparison to the Italian subspecies of honeybee. So there is, and even within subgroups, artificial selection by some labs or some researchers or beekeepers have been underway to increase disease resistance. So I'm working with Professor Chakmak at Uludağ University, and he has a bee line on Marmara Island that's been selected for Varroa resistance, for example. And we're interested in understanding the molecular mechanisms that are responsible for that. So the answer is, in summary, yes. Yeah.
Thank you for your answer. And the next question is there an inverse correlation between the health of bees and polluted air volume? How important this might be regarding infections?
Yeah, so we don't know because that hasn't been explicitly studied, and it's just now being recognized. My recent study that I did point this out, that pollution could be impacting bee health as well, and we haven't really. We always say, okay, use bee sentinels for monitoring ecosystem health, but we don't think about the health of the bees and how that's impacting them. If we find poor, say, air quality or something like that. So I would imagine it, negatively affects their health, but it really hasn't been explicitly investigated that much, looking at air pollution. It's a bit more of a concern now, I know, in California, for example, with the wildfires and all of the, the reduced air quality, and there's a lot of beekeepers in California. So that's something that might, I think, pick up an interest, I think. We had a lot of wildfires in Turkey too, and that really damaged a lot of beekeepers. And so the fires can damage air quality for quite some time. So that would be a concern, I think, for beekeepers. So I would imagine it would. I mean, they are aerobic organisms that require oxygen. Their flight is very metabolically expensive, so they need to burn a lot of energy to be able to sustain flight. And if they can't sustain flight out away from the hive, they're pretty much done for. They can't survive on their own. They're so social. They need their hive members to be able to survive. So that's an issue. Yeah, if they get stranded out away from the hive, then it becomes problematic for them. They'll probably die. So yeah, that would be interesting to see. Actually, is to focus on that instead of the pesticides, for example. It's a whole new dimension that could be impacting bee health.
Thank you for your answer. We have a couple of questions left.
Mm-hmm.
Different approaches for protecting the health of bee colonies has been studied. Are unpredictable climate change also considered during these studies?
It's been brought up as an issue. It hasn't been studied for honeybees explicitly that much because I think it's difficult to study. That's the main reason, but it's pretty widely accepted in the bee scientific community that climate change is likely to be a factor, in added stress. And in fact, some research has shown that weather is the strongest predictor of whether or not a bee colony will make it through the winter or not. And this past season in Turkey, it was very mild. It was actually a bad thing for the bees because they never went into hibernation mode, and they were continuing to try to forage and find flowers when there wasn't any flowers available because it was wintertime. So certainly, I think it does, but it hasn't really been measured that much directly, unfortunately.
Thank you. And the last question is, Einstein pointed out the bees' importance in the ecological environment by stating that mankind would not be able to survive without bees for more than 4 years. What are your opinions on this statement?
So I went to a bee conference and someone told me that this, a lot of people think it was Einstein, but it was actually a French philosopher that made this statement, this quote. I thought it was Einstein too, because it was reported that way. So that's one comment on it. But yeah, I think it might be. Well, so honeybees are they're generalists, so they visit a lot of different flowers, a lot of different flower species. I think, and then you have bees in general. So bees in general, there's about 20,000 different species, are very powerful pollinators, and that supports a lot of plant biodiversity, which aids in ecosystem stability because there's a lot of organisms dependent upon plants for food. So yeah, I can see it being like a significant destabilization cause to ecosystems. But I have a feeling it's maybe a little dramatic. We'd probably find a way to survive, but our life would be, I think, severely affected. Like we may be living off of like wind pollinated plants, like wheat, for example. So we might just be eating bread all the time to survive, But I think now we might be able to find a way to survive, but hopefully that won't happen. Hopefully we won't get to that point. But yeah, I mean, climate change is a huge issue, I think, that we need to address, not only for the bees, but just for humanity in general. So.
Thank you so much for all of these comprehensive answers. And you also shared such interesting facts. I'm sure our attendees have learned a lot.
Yeah, my pleasure. It was fun to give the presentation and to have so many questions. So I appreciate those. They're very nice questions.
Thank you very much for your participation. We have been really happy to see you among us.
Likewise. Thanks so much. Yeah, if there's any other questions, you can feel free to contact me, to email me. You can find my email on the internet easily, I'm sure. So.
Thank you.
Yeah.
So we wish you a healthy day, and we hope to see you again.
Sounds good. Thanks very much. Yeah, wishing you a good night, and yeah, thanks so much for the opportunity to present.
You're welcome. Dear attendees, thank you for your participation in BioInfo course. Events organized by BioInfo Orange platform will continue. You can follow all the announcements on our social media accounts, and you can also always contact with us. We wish you a healthy day.